So one of the questions that came up from one of the attendees was how long does the initiation, regeneration, and multiplication process take from the day you get the plant to when the client or the partner gets the plant back?
Okay, so the initiation first part, let's go step by step. Initiation is a step where we clean up the plant. So in case of initiation, it's normal procedure is you bring the plants, we clean this, wash this, take it in the initiation, and until the pre-multiplication, is if the plants is clean, it's a 3 months procedure to go to the complete phase of multiplication. However, if you receive a 3 to 6 months, if you receive a plants with lot of intercellular bacteria or intracellular bacteria, what happens if you do the cuttings out of the plants, you expose the plants quite a few times and keep cutting those mother plants again and again. So you open the wounds, and then the intercellular bacteria can enter, and then they can stay in the tissues. If it's the pathogenic bacteria, that you lose the plants right away. But there could be a non-pathogenic bacteria which is competing. So once we bring in the tissue culture facility, we need to wash, clean. We do the surface sterilization, but we cannot clean the inside. So what happened? The bacteria come back because media is highly nutritious, and then sugar is there. So they started coming out and causing some problem. There is a process of cleaning those intercellular bacteria, but it's required time. So that's why we usually say 3 to 9 months to do the initiation process. Multiplication depends how plants you need. You need 1,000 plants. Is very simple. You just produce in 2-3 months. You can produce 1,000 plus. If you need 1 million plants, you have to produce more. Say stock up to say 200,000 plants. You do stock. One multiplication you can make say 800,000. One multiplication one more time and do the elongation 1.6 million. You take 1 million out. Keep the 600,000. Do that one more transfer. So every month you can take it out. Depends on the demands, how much plants you can produce from each variety. And the rooting is generally 3 to 4 weeks in vitro rooting phase. Yeah.
Awesome. Sma, we had a slide on it in the beginning, but just to kind of jump back, and could you give a little bit of background on your experience in tissue culture and kind of how you got into running tissue culture for cannabis, and maybe some of the differences that are a little bit different compared to other crops you've worked with in the past?
Yeah, sure, I can explain. So the cannabis is an annual plant. So it is not a lot of tissue culture has been done is annual plant. That's a little bit different than regular hardy ornamentals or blueberries or apples or any other plants. But the process is same. You bring the meristem culture. You bring it in the tissue culture format to the multiplication, bringing in the tissue culture stage 2 and 3 and bring it out. I am working on in this crop almost like 20 years. With 15 to 20 years with the license under the federal license agreement here and there, but last few years I worked a lot in this area, and then we developed all these procedures, and we established all these stages, including the multiplication. We can actually drag the multiplication from 2 to 4 or even 6 times multiplication, depends your demand. We don't in this industry, in this variety, in this plant species, we don't want to drag these plants from high multiplication straight to the rooting. That's how you lose the plants a lot of time in the multiplication. We have to go through a stronger plants to produce in vitro and then do the rooting. That's the secret of these plants. So we have to be patient with these plants because it's an annual plants.
Awesome. Carson, do you want to take the next one, or do you want me to launch off a couple more questions?
Yeah, sure. We had an interesting one come in from identifying what the meristem looks like in cannabis, and specifically, the 2 set of small leaves showing at the internode, but what do they look to identify when actually obtaining the meristem?
Meristems are like a dome shape. If you cover this, if you think about a cabbage, you take this cover out, you can eventually get a small dome-shaped structure. So that's the meristem. So yeah, I'm just giving an example. In this case, you don't have to do, they don't have that many layer of leaves. So after few leaves in that very top, easy for using a stereo microscope and to take uncover those leaves, and you can see a dome shape, very small part. You need to isolate that one. Is very tricky work. You need to isolate that part and wash, or you can do the wash first and isolate them, and then you putting in the test tube. They start growing because these are actively growing cells. Cell division is very fast. They're very fast. They grow.
Awesome, thank you.
So we've had a couple questions about clonal variation, so I'll try to kind of combine them and then fire it off in a two-parter. But you know, I guess to start off, how do you identify clonal variation if it does occur? Do you have to flower the plant directly, or is there an easier way to be able to screen it earlier?
Yeah, easiest way to screening earlier is the doing the DNA fingerprinting, which we are doing. Sometimes, unless you take the plants out, grow them, and find out that they are producing something different than you expecting, you probably cannot say. Sometimes say it's clonal variation or not. However, in the tissue culture, sometimes you can see, if you're familiar with your plants, you can see some plants are looks different. Either they are not doing good or not doing too good with your regular batches. So sometimes you can see, but it's very rare because sometimes plant genetically same, but due to different reason, they could be behave, one transfer may behave different. But if it's consistently behaving different phenotype, then you should isolate them and send for DNA fingerprinting rather than waiting and like taking all the way to the grow the plants and see. You should send it for DNA fingerprinting and see that it is the same, still same as the original one. In cannabis industries, cannabis plants came from so many breeding program, from so many breeding has been done. So there is always a possibility of sometimes they want to go back to one of their parents. So there is a possibility that that could be a mutation happening. But by using a proper technology in the tissue culture, we can avoid the clonal variation. And we are currently doing in Segra tissue culture facilities, so avoiding all the possible chance of clonal variation, and that's how we are actually screening also every 2-3 months to see the plants are still stable or not.
And then kind of a quick follow-up question, and thanks for that. That was a good good explanation. But you know, kind of a quick follow-up on that. Is clonal variation the same term? You know, is it synonymous with genetic drift, or are those two different terms? Are you able to kind of clarify the difference there?
It is clonal variation. It can go either way. It can be theoretically it is through the tissue culture. It is changing their mutation, either good, bad. Clonal variation could be good. Sometimes it's used in the breeding program. You can isolate a plants and then you can separate that one. If the plants are doing, it could be a better crop, their original one. It could be worse crop. We don't know. Genetic drift, to me, it could be drift in generally. It's like lowering the lower production, lowering the food crop production, lowering the growth. So it generally reduce the vigor of the plants and the food production. So we don't know why the genetic drift happen. Genetic drift could be just a result of, because we are doing conventional cutting, the wounds are opening again and again. So intercellular bacteria, which could be non-pathogenic, or virus, which could be non-pathogenic. That's why you cannot see any symptom, but they are competing the food with the plants itself. So plants is producing food. They are taking the share from all these microbes inside. They're taking the shares. And that's how they are slowly performing poor. That could be that one. There's further research is required in the genetic drift, the term, and then explaining why it's happening. And as Segra's earlier, one of the publications is showing that genetic drift all happening, but their genetics, the DNA is not changing. They are still same, but they're poor performance. So further research is required for the genetic drift. But I would say Sma clonal variation is completely different than genetic drift.
Awesome. Thank you. Thank you.
We've had a couple questions based on the actual media that you use for tissue culture. Can you comment on the hormones that you use for multiplication?
I cannot directly explain what kind of hormone. This is, I think, a Segra trade secret. I'm not, but I can in generally say we use the cytokinin ratio of cytokinin and auxin, and we usually we develop these protocols by combination of different auxin and cytokinin with different nutrient supplements, and we established 3 to 4 different growing media for multiplication phase, and 1 for initiation. So some varieties, when they come, they all go through the first one, and then they for initiation they transfer to priority 1, 2, 3, wherever they should. If none of these produce good enough, then we have to, those plants go straight for the research team, and they develop another protocol. We try to avoid, even we have a lower multiplication, we try to avoid induce so many different media, because commercially it's really difficult to produce small batches of media. We try to give them first, second, or third, if possible, if even with a lower multiplication, so we can make a bigger batch of media and save the labors. I hope I answered your question.
No, that's perfect. I appreciate you going into as much detail as you can there.
You know, another one that came up that's pretty interesting, it's kind of come up a couple of times, is what are common challenges you see associated with groups kind of trying to do tissue culture? You know, as they run through the process, where do you see the most common kind of areas of struggle for groups trying to initiate and multiply cannabis plantlets?
One is the well, first one is, I would say there is always chance of receiving contaminated plants, because this industry is hidden so many years. People are using mother plants from mother plants, and they are using the cutting, and then intercellular bacteria. I receive so many plants which is loaded with intercellular bacteria. So initiation take longer times. It's not that I sometimes I receive a plants which in a month they are really good. In 3 months time we start producing the stage 2 or stage 3. We receive that kind of plants also, but most of the cases they are compared with horticulture industry, there is a lot of intercellular bacteria we need to clean. And one of the places we noticed that when we gave these clean plants, they compared with their original cutting plant, their original cutting is took for me to produce that plants in almost 9 months. And when the new plants from tissue culture came out, it looked like completely different plants. The vigor was so good, plants were so happy, they're producing more fruits, and looks like completely different plants to them. So this is one of the biggest challenge we are facing. But when all the cleaning is done, it's really good plants coming out, and it's done. So this is the only biggest challenge we are facing.
So essentially just, you know, systemic pathogens built up through years of traditional cloning, and just taking the time to get rid of those, and then kind of tweak multiplication rates for each cultivar.
Yeah.
Awesome.
Interesting. We had a interesting one come in here saying that so there was a theft of a root ball from the dumpster outside of an LP. Now, could tissue culture be used to regenerate this into more plants using, I guess, undifferentiated cells in that case?
Yeah, with that, from your question, I believe is you asking that is it possible from regenerate from tissues of roots or any other things? Yes, theoretically, yes, you can produce. That's we call indirect regeneration. So in this case, you don't have any meristem. There is a chance of virus there. Also, you do the inducing indirect regeneration, which there is a possibility of mutation again. But once you screen you regenerate these plants, you screen those, see the DNA test and see the virus test. There is some plants obviously will hopefully be clean and true to type. So we select those plants, even a 5-10 plantlets, clean and true to type, and rest we can destroy, and we can use those for multiplication purpose and produce a bigger batch. Yes, possible.
Possible. Okay, interesting. Well, let's hope that's not the case in that situation then.
Yeah.
Awesome. Another one back to the kind of the DNA fingerprinting. Do you DNA fingerprint every plant before it goes to a customer? Is that kind of standard SOP for anything leaving the facility?
We don't do every plant. We do the regular auditing for each batch. And generally, if a bigger batch, every 50 plants, we take one sample. And every 2 to 3 months, we do the repeat the program and see any chance of variation happening. When everything clean is certified, and then we do put it in the stage 3. In that cases, we do we have a 3.5 vessel. We do the every vessel. So one vessel usually have almost 100 plants or 90 plants. So we do each of these vessel one sample test and see any DNA any change happening. If any of these have showed any kind of abnormality, we don't send the whole batch. Usually stopped. Further investigate, we go back and do each plants. In that cases, select say 50 or 50 plants. We select, okay, these are the select. So 87 came, 47 came good. We keep the 47, rest 3, and rest of the whole population has been destroyed. And then we go back and try multiply from only those 47. If the repeating happening like this, and we go back to the mother plant and do the reinitiation.
Awesome. So essentially, we test every batch before it leaves, but throughout the multiplication process, you're doing testing along the way to track any changes before they kind of expand and get to that final batch also.
Exactly. Yeah.
Fantastic.
Awesome. We have another one about flowering cannabis plants. Can you take a plant that's in flower and initiate it back into tissue culture, back to vegetative growth effectively?
Answer is yes. We just developed a technology where we can actually regenerate from the flower, immature flower bud. Again, the process is that we also test the DNA fingerprinting for true to type or not, and also we do the virus test if possible. A pathogen test is there any virus available in these cases, because these are non-meristematic tissue culture. Yes, we can do from flower buds.
And does that process generally take longer than it would if it was in vegetative growth?
Not necessarily. If it take longer, if the screening probably add 1 more month, but the regeneration is same as the meristem. Meristem actually take longer time, but these are the bigger flower buds. They multiply very fast. So at the end, probably same time, similar time.
And Sma, is that just kind of like week 1, week 2, kind of early flower, or can you also do that, with late flower plants also? Or is it just
we haven't tried any late flower. My guess is late flower will not be possible because their oxygen level is really low in that cases. Yeah. So my guess will be we haven't tried with the flower, mature flower, but it's immature flower and flower bud is possible.
So try to get it in earlier rather than later if you're going to send something in.
Yeah.
Awesome. Another one pretty interesting about kind of pathogen resistance and re-exposure once it's cleaned up through tissue culture. Once the plants get back into the facility, and they've been cleaned up, but they might be re-exposed to powdery mildew or other pathogens in the facility. Do you have to re-clean them, or is there kind of some inherent disease resistance built up through tissue culture also? Does getting rid of some of those systemic pathogens bolster resistance for future exposure also?
No. So answer is no. You cannot develop a disease resistance through tissue culture. You have to, this is different part. It's a genetic engineering. You have to develop breeding, develop a disease resistance. Tissue culture, what it do is that they scale up this production. So in another way I can explain, if you bring a plant from tissue culture exposed to the environment, those plants can fight back better than your original plants because those are healthier plants clean plants, and they have better immune system, so they can fight back and develop. But eventually, if you take the cutting out of these plants again and again and again, it will be same as mother plants, so they lose their ability. So it's better in agriculture, horticulture industry. We always do the ask request for the every time the new batch from tissue culture. So you get a clean batch all the time, and they will resist for time being, and they will resist any powdery mildew or pathogen. We check these tissue culture plants. One of the facility have all invested with a lot of powdery mildew. Only plants survive were the tissue culture plants and the first generation tissue culture plants. We proved that one already. So ideally, yes, we should have a tissue culture batch, fresh batch coming from all the time. So you have always clean plants coming.
So essentially, you know, it'll be able to fight diseases and pathogens off a little better because it's starting out clean and starting out a little bit more vigorous.
Yes,
but,
it's much better, yes.
But, you know, to really get a fresh start at the end of that cycle, the best way to do it is to kind of get new tissue culture plants for flowering, or just tissue culture mothers to kind of start out with a fresh, clean plant.
Yeah, so, exactly.
Awesome. And I think, Carson, do you want to launch off? I think we probably have time for about one more.
Yeah. There was one about so when we send plants out in the micro plug rooted there, what do they have to do in order to get it ready to be in the field or into the vegetative growth there?
Okay, so the micro plugs we are sending, they're already rooted. All have to do maybe very quick acclimatized to their environment, 3 to 5 days. So once the plants are acclimatized, they can go to the veg room. So the plant can be transplanted directly, depends on the what size. And then it can be directly planted. The picture, the slide currently showing the left side, the plug, they're already rooted and everything. So those plugs straight can go to the, after acclimation, straight go to their veggie production facilities. Generally, we recommend that this plug, wherever they go, is straight go insert in the bigger pot or loose filling or a plug with a hole there. They can just fit it, and then the root can go through their own way, and then they can establish. Very simple procedure, very convenient. You receive the plants, you just acclimatize and take it to your places, and then you start growing. You don't have any hassle for cuttings or going through all these pathogen attack and everything. Is like clean plants, receive, acclimatize, plant it. Very simple.
Awesome. And can you comment quickly on just what the composition of the plug is?
This composition is a mix of cocoa and I believe cocoa and perlite. So it's has this ability to absorb water, and also all the required nutrients we are also added here for shipping and staying there.
And they're all,
thank you.
Are they all clean before they
all, yeah, the plugs are sterile. When we started with, everything is sterile. Remember, it's tissue culture also. We do either currently we are autoclaving, but eventually we'll do the gamma ray for larger scale for each plug, and then planted there. And once they come out, they can shipping time they're exposed to the outer environment because it's not 100% sealed, but it's they, as I mentioned, they can fight back because they are cleaner plants for pathogen or anything.
Awesome. Well, I think we probably have to wrap it up here, but thank you all so much for the interest there. I know there are a few more questions that we didn't get to, but we'll follow up with everyone and kind of help answer those as one-offs. Please feel free to reach out to Carson or me directly. We'd be thrilled to chat more about tissue culture and discuss how it may be able to improve productivity or mitigate risk within your facility, either with your cultivars or trying some new ones of ours.